and growth hormones which are required for the growth of the plant, that are auxins and gibberellins, which are very much essential to grow any plant. So the plant that we selected for the study is not much known about this plant in Karnataka. It is called Sauropus androgynous. It is commonly called Chakramuni. In Karnataka it is called Chakramuni, and then in Tamil Nadu and Andhra Pradesh they use it Chakarmeni. It is also called multivitamin plant. I will tell you later on about this. And the classification is division. It belongs to division Magnoliaphyta, Magnoliapsida, order Malpighiales, and it belongs to family. Now it is put in the family Phyllantheraceae. Earlier it was in the family Euphorbiaceae. And the common names include, because of its flower shape, it's also called star gooseberry and tropical asparagus. In Thailand, especially in Thailand, there are shoot tips. It is used in making salad, and the leaves are very palatable. The taste is very rich, and then it gives a taste of freshly harvested garden pea. I have tasted, and it is very nice to taste. Raw leaves we can eat. And then it is also called as sweet leaf bush. And then in Thailand it is a very common vegetable, green vegetable, that is called pakwan. And this is the plant. You can see the plant and then the flowers. And as a shrub it grows more than 2.5 meters. The leaves are dark green and oval in shape, and just the leaves are 5 to 6 centimeters long. And it's a most popular leafy vegetable in South Asia and Southeast Asia. It's not originated in India. There is no proof, but they say that it must have come to India by maybe near 1950s from maybe Thailand or Malaysia, because it's a popular vegetable over there in Thailand, Malaysia, and then even Indonesia also. And it is famous for its palatability and nutty taste. And another important thing about is it contains the alkaloid papaverine, and it can be propagated through stem cuttings. This is the best method, that is stem cutting, as the seed longevity is poor, and stem cutting is the best method of propagation of this plant. And another important thing about this plant is it is called a multivitamin plant because it is rich in vitamin A, B vitamins, and then vitamin C, vitamin E, and it is one of the few plant sources which contain vitamin K, and it is also rich in proteins and minerals. And the medicinal property. See, this plant is a medicinal plant. At the same time, we are consuming it as a food, one of the green vegetables. And I have told you, as it is rich in vitamins, it can be used to cure vitamin deficiency. Just in the morning after breakfast, you can just pop up 2 or 3 leaves if it is growing in our pots, and then after just 30 to 45 days you will get rid of this vitamin deficiency without taking any vitamin tablets. And then it is antipyretic, and it can be used. In Thailand, most of the plants that the medicinal properties have been analyzed. It's even now used to treat mumps and general body weakness, urinary problems. The leaf extract can be used to cure earache. And coming to the materials and methods, we have used biofertilizers like Azosphaera, which is a very good free nitrogen fixer, to mobilize nitrogen to the plant so that we can reduce chemical nitrogen manure like urea. Because to grow, if we have to use the leaves, then we need nitrogen manure. Nitrogen is more important for the growth of the leaves. So this is a very good nitrogen fixer. It's a gram-negative motile bacteria belonging to the order Rhodospirales, and then a phosphorus solubilizer, a phosphate solubilizer, that is Bacillus megateria. It's a rod-shaped gram-positive endospore forming bacteria, and it is the largest eubacteria found in soil. And then we procured the plant Sauropus androgynous from Dhanvantari Vana, that is in Nandavanti campus. And then the biofertilizers we got it from Horticulture Department, Government of Karnataka, from Kodimavu. And then the treatments, we gave treatments to these plants in pot trials, and then that is the T1 azospirillum, T2 Bacillus megaterium, T3 vermicompost, T4. Just we tried the combination of all the three. If we can we get a better result using all the three, and then we compared it with control without adding anything. And then we did nutrient analysis by using that is nitrogen by Michael Jerdal method, and then phosphate by Jackson's method, and potassium by Toth and Prince, because as we are giving the biofertilizer that is which can fix nitrogen or mobilize nitrogen and phosphorus, we need to estimate how much it has taken up, that is the nitrogen and NPK. And the results, this is the 60 days old plant, you can see. This is the 4 treatments along with the control. You can see T4 is giving a better result, that is at 60 days. Next is 90 days old plants at which we did all the analysis work. And this is what we are doing now. It is, we are doing on the field near Kanpura Road, that is after pot trials we are doing on under field conditions also. And the growth parameters showed that a combination of treatment, that is azospirillum, PSM and vermicompost, gave a better plant height and number of leaves, because here leaves are more important, and this is a better combination compared to the single inoculations. The combined inoculation gave better results. But NPK content, when we studied, there was we didn't get the first priority, that maximum we didn't see in the combined treatment, but in for nitrogen, vermicompost treated plants, maximum nitrogen content was there, and then followed by the combined inoculation. And for phosphorus, Bacillus megaterium, as it is a phosphorus solubilizer, gave a better, the first maximum content, P content was observed. And then for potassium also, vermicompost gave a better result. That is, of all the three, vermicompost was better when we just analyzed NPK content. But it was followed always by the combination treatment, that is all the combined treatment. And then finally, to conclude, as we use the leaves for our as a vegetable and also medicinal purpose, the best combination is combination of all the three proved to be the best for the plant Saurochus androgynous. And this is a part of the minor research project which was granted by UGC University Grants Commission for myself and Dr. Pratibha. I am the principal investigator and Dr. Pratibha is the co-investigator, and I thank UGC and our college principal, Mahatma Science College, and our department and Department of Botany, Bangalore University, and the organizers of this conference who have given this opportunity to present my paper. Thank you.
Mentioned all the vitamins and minerals are present in the leaves.
Yeah, the paper, I will give you the paper.
So
I haven't done references
data, because
there.
it shows that in the particular plant the leaves contain all vitamins and minerals. In that case it should be patented.
No,
Okay?
it's not
It
patented.
is directly can be consumed without food also. People are the addition of vitamins.
That is right.
Vedas times
Artificial name plant. I have a paper. For your reference, I'll give. I have brought that one also. Thailand scientists have worked on that. And we people were not knowing. Another important thing about this is that papaverine is present. No, it's not very good for our consumption. It sometimes it will make some food poisoning also. There is a negative effect, and it should be consumed in a very small quantity, not.
Since it is branded as a medicinal plant,
Yeah, yeah.
the application should be only for.
Yeah, now it is gained. Actually, I didn't take much references because not much work has been done on this. And as we are teachers, not much room, especially for graduate college, degree college teachers for doing research and all, not much time is available for us to do. So we are just started
If
and
we
we are
have the
doing it.
quantification of all the
yeah, we are
vitamins,
doing.
then we can say that the antipyretic activities,
Yes.
particularly antioxidant activities,
Antioxidant, very high antioxidant
Leading to the-
activities there.
that means what I can say, that the skin disease control and all these things can be applied.
Yes, and then we are doing it, sir. It's underway. One more year we have for the completion of our project. I showed that land picture, no? From that we are doing all the quantification, vitamin analysis, title of the project itself is antioxidant analysis of the medicinal plants. So we are doing that one, and we have references also. So I will show you that reference. Some Thailand scientists have done that. And maybe another 6 months we will be finalizing our results. Thank you. Thanks a lot.
Then outcrossing and inbreeding, it is possible to establish a homozygous refractory strain, and thus these strains could be used in the genetic control program. So the resistant strains are positive and they play a positive role in evolution. Next. Now other changes, what are the other changes? The reproductive changes. The invertebrate reproductive biologists are interested to know that if the insecticide resistant strain, what are the changes that are taking place in the fertility, fecundity, hatchability, etc., etc. The fitness of the population is very important. The fitness, gene for fitness plays a very important role in speciation and evolution. And so we have done the fecundity, hatchability, sex ratio, male reproductive system we have done. This was done mainly to see the better understanding of biology of insect. Variability in the reproductive traits have important consequence for any control strategies or creating transient strains, understanding the process of speciation. Next. Now these are some of the insecticides initially that have been used to study this process. Next. There was a decrease in fecundity, hatchability and sex ratio distortion towards males were observed in all the above said insecticides. Male accessory glands were reduced in size, testes were small, rudimentary atrophied, walk differences were thin and disintegrated. Rudimentary atrophied testes were the common feature of all the insecticide resistant strains. So here you can see normal one, and here you can study to vast difference. Here you can see the changes. That means it has impact on the reproductive system. Next. So insecticide resistance genes and speciation. So the insecticide resistance genes plays a positive role in speciation or the evolution of the species. And therefore in this present lecture we have compared the insecticide. First of all, we have established the genetic basis of insecticide resistance of most of the insecticides that are available in nature, and then studied the genetics of these, and then established multiple marker for resistance, and try to synthesize a homozygous strain and see how best they can be used in synthesizing transgenic strains. Biochemical studies were also carried out by using all the stages of mosquito, eggs, larvae, pupa and adult, and then try to use it as an indicator or marker to test the rapid technique to identify the resistance, and thereby effective control programs can be undertaken by the public health workers. Cytological.
Thank you, sir. Now the audience can interact with the speaker.
In the backdrop of the insects developing resistance against insecticides, including the mosquitoes, is it not advisable to go in for pesticide cocktails? Because that's one of the ways suggested to overcome the pesticide resistance or insecticide resistance. Insecticides together, it's less likely that the insects would develop resistance against 2 or 3 insecticides used together. That's one of the ways. Second thing, in order to, because we are discussing on a very important aspect in the present conference, that the environmental protection is of utmost importance from the use of toxic chemicals. Biocontrol approach is one of the ways to tackle the problem of pests. Even in mosquito, we can as well go in for the use of some of the bacterial pathogens like Bacillus thuringiensis and also metabolite organisms. They can also be used as biocontrol agents because this is only to tide over the problems posed by pesticides. So will you please highlight all these things?
No, that's why you see specific insecticide which you apply and then you change the insecticide, they develop multiple resistance. And agriculture pests also develop resistance. The same phenomenon occurs. Sooner or later, every species develop resistance for insecticide. And at this stage, it is not possible to control the insects or any pest by applying chemicals. So therefore, we have come out with the novel idea. You develop a genetic method where it is autocidal control. It is specific only to the target species. You produce the strains in the laboratory and produce them hundreds and millions in the laboratory, release them into the field. Only the males will be released, and thereby it can suppress the population. So this is the method which has been recommended by WHO and other international agencies. And we in our laboratory, even in the early 80s and late 80s, we produced all these strains and we are in a position to release them in the natural population. Because of some reason, we did not do that. Apparently, it's a very, very difficult situation indeed. It's very difficult and only few people can do that because it involves a lot of genetics, a lot of work it is involved. But by using this method, the people in Europe and also some parts of Southern America, they are successfully used, even in Africa, they are successfully used. And therefore, the alternative strategy, which does not involve in the pollution of the environment. The environment should not be polluted, the non-target organism should not be killed. The most important thing is non-target organism should not be killed, both beneficial and harmful. With the anxiety of killing a particular vector species, we are killing the beneficial insect also. So this should not be done. And secondly, environmental pollution is alarming situation. This should not be done. So therefore, alternative to this, alternative to the insecticide, you have to develop other strategy. The genetic control is the most effective method which could be used. But for some reasons or other in India we are not using. But in the Western world, see, people are using that. As a Humboldt fellow in Germany, 2 years of my stay, we used a particular strain of Aedes aegypti to Aedes aegypti in pilot release experiment. Actually, I was also mainly involved in that, and we could control this population, this species, within 2 years, see, by applying by releasing the translocation spear. So likewise, you see, we had a lot of system we had. So we have to develop alternative way. As you said, syringes or biological control or larvicide fish, anything and everything. Most importantly, the environment should be maintained. The management of the environment is very, very, very important. Whatever method you apply, management of the environment, which people are doing from millions and millions of years back, they are saying that proper drainage should be there, cleanliness should be there. So if you maintain the environment, management of the environment, see there is no free water, and you see that mosquitoes will not lay eggs on the water, they will not multiply, and see that the mosquito population is reduced. Therefore, it is mandatory, it is important at this juncture to develop an alternative strategy. Sometimes it may not be complicated, it is just a simple method, and each one of us can maintain your own environment and see that there is no water or seepage everywhere. And you construct a house, there is a lot of water. In urban, you see, everywhere you construct a house, you have water. And make sure that water is not used for the mosquitoes for laying eggs, and thereby the mosquitoes are coming out. So this is very, very important. So now the most important thing, as you rightly said, that managing the environment is the most important thing, and this could be done by the cooperation of everybody. See, the government is there, public health department is there to control this. This is secondary. Now look at this in our country. 90% of the budget is spent on controlling the mosquito-borne diseases, dengue, malaria, and all that. See, each one of us will maintain our environment properly. You can, to some extent, to a large extent, you can control that. So the different mechanism, as I strongly advocate that the genetic control is one of the alternative strategy, because agriculture pest at the international Atomic Energy Agency, see, they are using this sterile insect by using translocations. They have successfully controlled in many parts of Europe and South America. And mosquitoes also been controlled in Africa. And in many places, my own professor, he did the first successful experiment in the late 60s and 70s. He demonstrated the genetic control mechanism in the late 60s and 70s so effectively as proved. So it should have a political will. There are different insecticides. You see, it's a big business.
No more.
Webs, they are wanderers. And family Ericidae, these are known as social spiders because you can see large number of spiders in a single web. Up to 100 to 150 spiders of different ages coexist in the same web. They construct something known as a sheet web. And one characteristic feature here is they do not kill their prey. Usually spiders first inject the digestive juices into their prey and then consume them. But these members, they eat their prey when they are still alive. And all the members of the colony share that particular prey. These are also known as two-tailed spiders because of the presence of the posterior spinnerets, which are extremely long. The spinnerets are sometimes even longer than the length of the total abdomen. So that is what gives the appearance of a tail. These spin cocoon webs, not usually, but only when they are about to lay their eggs, and they preserve their eggs inside the cocoon web. And family Lycosidae, they are also known as wolf spiders because they run down their prey.